Hi all. Today we are discussing about the reassociation kinetics of unique DNA. We will also discuss about the topics moderately repetitive and highly repetitive DNA, different types of DNA, COT value and complexity of genome. So the reassociation kinetics is somewhat related to genome. The genome is the sum total of all genetic material of an organism. Amount of DNA present in genome of a species is called C value. In case of lower eukaryotes, genome size increases with complexity. In higher eukaryotes, there is no correlation. This lack of correlation is known as C value paradox. So, what is reassociation kinetics? The reassociation kinetics is a technique that measures how quickly complementary strands of DNA reassociate. It can be used to measure the complexity of a DNA sequence, including unique DNA sequences. The rate of renaturation of fully denatured DNA is kinetically a second order reaction. So, we can discuss about the features of reassociation kinetics. The first one is rate of reassociation. The rate of reassociation is the rate at which complementary strands encounter each other. Second one, the concentration of complementary strands. The higher the concentration, the faster the DNA will reassociate. So the concentration of complementary strand is highly related with the reassociation. Next one is rate constant. The rate constant is inversely proportional to sequence complexity. The fourth one, time to half renaturation. The time it takes for DNA to renaturate halfway is directly proportional to sequence complexity. So, there are two components of reassociation kinetics. They are DNA fragmentation and dissociation. So, the DNA fragmentation is the DNA is cut into pieces that are hundreds of base pairs long. The dissociation is the DNA is heated to break it into single strands. And this method is processed in two ways, the denaturation and renaturation. So what is denaturation? Denaturation is the process by which the loss of helical structure of DNA into single strands through heat. In laboratory, the DNA strands can be separated by change in pH, heat. These are the two components which can separate the DNA strands. Denaturation is measured by spectrophotometer. The melting temperature of DNA. The melting temperature of DNA is described by Tm. It is the process by which the temperature at which one half of DNA duplex will dissociate to become single-stranded is called melting temperature. So what is renaturation? Renaturation is also known as annealing. Separated complementary strands of DNA can spontaneously reassociate. By cooling to form double helix is known as renaturation. Renaturation led to the discovery of repetitive DNA. The temperature of DNA lowered below its melting temperature. The rewinding of DNA takes place. DNA can melt and re-anneal itself reversibly. So the experiment is conducted by three steps. The first one is the double-stranded DNA is heated. This heating results in the denaturation of DNA. So the DNA which is heated is slowly subjected to cooling and solution is now cooled and the DNA fragments reassociate called renaturation. This is the process of renaturation. The time required for half renaturation is inversely proportional to the rate constant. Let C is equal to concentration of single-stranded DNA at time t. The rate of loss of single-stranded DNA during renaturation is given by the following expression for a second-order rate process. Minus dC by dt is equal to Kc square. By the integration and some algebraic substitution, it shows that the equation is subjected to C by C naught is equal to 1 by 1 plus Kc naught T. So here the C represents the concentration of single stranded DNA after time T, and C naught is the initial concentration of single stranded DNA. Here the K is the second order rate constant and C naught T is measured in moles or liter. Repetitive DNA. The repetitive DNA is classified into three: non-repetitive DNA, moderately repetitive DNA and highly repetitive DNA. The non-repetitive sequence that are unique, and second, the moderatively repetitive sequences that are dispersed and repeated a small number of times with some copies not being identical. The third one, highly repetitive sequences which are short and usually repeated more in number. So the repetitive DNA is found almost in all eukaryotes except the yeast, but almost negligible in prokaryotes. Such DNA is found near the centromere in the chromosome. There are multiple classes of repetitive DNA. Two of these classes include So next we are coming about the repetitive DNA. The repetitive DNA is found almost in all eukaryotes except yeast, but almost negligible 0.3% in prokaryotes. Such DNA is found near the centromere in the chromosome. There are multiple classes of repetitive DNA, and the two classifications are highly repetitive DNA and moderately repetitive DNA. The repetitive DNA is essential for genome function. Generic repeated signals in the DNA are necessary for genome replication and accurate transmission to progeny. Highly repetitive DNA. Highly repetitive DNA sequence or satellite DNA consists of very short sequences that are present many thousands of times in the genome. Some of the highly repetitive DNA is clustered in structural regions of chromosomes, particularly in the centromic and telomeric regions. It is transcriptionally inactive. Moderately repetitive DNA. Moderately repetitive DNA consists of relatively short sequences that are repeated typically 10 to 1000 types in the genomes. Moderately repetitive DNA can be clustered at specific chromosomal locations or distributed throughout the genome. It is responsible for the high degree of secondary structure formation in the pre-mRNA. Most types of moderately repetitive DNA are short, about 300 base pairs in length, are interspersed with unique sequences, are often transcribed but do not code for gene product. The next one is satellite DNA. It is a highly repetitive DNA sequence, mainly found in centromere and telomere of the heterochromatin. It consists of short sequence about 5 to 5000 BP in length. It forms very large clusters in discrete areas such as centromere. Each cluster contains several million base pairs of DNA. In many species, the base composition of these DNA segments is sufficiently different from bulk of DNA that fragments containing sequence can be separated into a distinct satellite band during density gradient centrifugation. Satellite DNAs tend to evolve very rapidly, causing sequence of these genomic elements vary even between two closely related species. The junk DNA. Junk DNA or non-coding DNA sequences are components of an organism's DNA that do not encode proteins or polypeptides. Some non-coding DNA is transcribed into functional non-coding RNA molecules. Example, the transfer RNA, ribosomal RNA and regulatory RNAs. Other functions of non-coding DNA include the transcriptional and translational regulation of protein coding sequences, scaffold attachment regions, origins of DNA replication, centromeres and telomeres. The another type of DNA is selfish DNA. The selfish DNA is a term for DNA sequences that replicate within the genome of another organism, prioritizing its own interest over the host DNA. Selfish DNA can spread through a population faster than expected, even if it doesn't provide a selective advantage to the organism. So how does selfish DNA works? Selfish DNA multiplies within the host DNA molecule. It can spread through a population faster than expected. It can enhance its own transmission at the expense of other genes in the genome. It can have no positive or a net negative effect on organism fitness. Cot value. It is developed by Roy Britton and Eric Davidson in 1960. It is a technique for measuring complexity, the genome complexity. Based on principle of DNA renaturation kinetics, it also gives the percentage of single stranded DNA present after the denaturation. During renaturation, there are repetitive as well as unique sequences in DNA. So the principle of Cot value is rate of renaturation is directly proportional to the concentration of complementary sequences. This is the graph which shows the highly repetitive DNA, moderately repetitive DNA and single sequence. Genome complexity. So we can describe the genome complexity as the total length of different sequences of the DNA. It is measured through renaturation kinetics of denatured DNA. So the genome complexity is a measure of the relative amounts of unique and repeated DNA sequences in an organism's genome. It is a fundamental structural property of the genome. So the genome complexity measure can be done in three ways: the Cot analysis, renaturation kinetic experiments and the region complexity. In Cot analysis measures the total length of unique sequences in a genome in base pairs. Renaturation kinetic experiments measures the physical characteristics of genes and extra DNA. The region complexity counts the number of distinct random walks in a subgraph of a genome. The complexity of eukaryotic genomes. Genomes of the most eukaryotes are larger and more complex than those of prokaryotes. However, the genome size of many eukaryotes does not appear to be related to generic complexity. The much of the complexity of eukaryotic genomes thus results from the abundance of several different types of non-coding sequences, which constitute most of the DNA of higher eukaryotic cells. The more complex the organism, the more DNA is needed to run it. That is, the total amount of DNA in the genome. Therefore, we would expect a linear relationship between genome size and organism complexity. At the lower range of complexity, this holds. In larger organisms, relationship breaks down. The bacteria have smaller genomes than eukaryotes, and viruses have smaller genomes than bacteria. This is the C-value paradox, that in the most complex organisms, there doesn't appear to be the expected relationship between complexity and genome size. Thank you.